Thank you, Cameron, and thanks to the organizers of the D3 meeting of the International Society of Tropical Medicine and Hygiene, neglected tropical diseases. What meeting am I at? For the invitation to speak to you all today. It's a pleasure to be here. This is going to be a departure, a significant departure from some of the other talks in the meeting in two ways. First of all, I will not be talking about a complex cellular or multicellular parasite, but instead talking about a teeny tiny virus, albeit with its own inherent complexities. Secondly, I will not be talking about early drug discovery, lead identification, optimization. Instead, I'm going to be talking about phase 1 to 3 clinical trials of a couple of different dengue vaccine candidates. As Cameron alluded to, I was CEO of Envirogen, which was a biotech company that was focused on developing vaccines for emerging infectious diseases, including dengue, hand, foot, and mouth disease, and chikungunya. And based on our clinical trial results, then we were acquired by Takeda. And since then, I've been consulting with a number of vaccine companies, including still Takeda. So that's what I do these days. But on to dengue fever. Dengue fever, of course, is a neglected tropical disease, and it is a threat to global health worldwide. About half the world's population live in dengue endemic countries. It's been estimated that 390 million infections of dengue occur every year. Now, once you're infected with dengue, though, you can have a whole spectrum of disease, from asymptomatic dengue infection to dengue fever, also called breakbone fever because of the severe arthralgia or myalgia associated with the infection. And that's debilitating enough. But worse yet, then, is the fact that it can also cause mortality in the form of dengue hemorrhagic fever or dengue shock syndrome. It's estimated then, as I said, that about 390 million infections, about almost 100 million dengue fever cases, clinical disease caused every year, about 2 million cases of the more severe forms that can lead to, roughly over 20,000 deaths per year. Dengue is a small RNA virus. It's an enveloped RNA virus, about 11 kilobases. It is transmitted by the mosquito Aedes aegypti. From human to mosquito to human, there are for dengue, there are a few other perhaps primates, but often not, mostly just a human host and a mosquito host. The complications with dengue is that there are 4 different viruses that can cause the disease, creatively called dengue virus 1 through 4. Infection with one of those dengue viruses can lead to lifelong protection once you survive the fever or asymptomatic disease, or worse yet, severe consequences. You're protected for life from that dengue virus. The problem is if you get infected with a second dengue virus. And infection with a second heterotypic virus leaves you at higher risk of more severe disease. So for that reason, it's been difficult to develop a vaccine because you have to have a vaccine that simultaneously protects against all 4. Likewise, there currently aren't any therapeutic options other than intensive care for the severe disease forms of dengue. Thus, there's limited that can be done now, except intensive care does reduce the mortality but doesn't eliminate it. In terms of its worldwide distribution, this just shows a map. Essentially, wherever you find Aedes aegypti, you find dengue. Part of this is due to the fact that humans then move dengue around the world quite effectively, and then mosquitoes transmit it locally. So dengue is, of course, Aedes aegypti, rather, is a tropical or subtropical mosquito. It can't survive in temperate zones and cold temperatures. So it does do quite well then, limited by the isotherms throughout Central and South America and Africa and predominantly in Southeast Asia as well. Of course, with global warming, those isotherms could also shift, and which may also cause consequences in other regions of the globe. Because of its worldwide spread and its significant impact on public health, the WHO has a specific objective around dengue, which is to reduce mortality and morbidity from dengue in 2020 by at least 50% and 25% respectively. And of course, having a vaccine available would be a very effective means of doing this. It's one thing to show pictures of viruses and show pictures of worldwide distribution, but in fact, one of the quotes that we've heard in Bolivia during an outbreak was a father just saying, "I'm just watching my son die slowly as we knock on different hospital doors." It's a very frightening disease for parents and obviously for children as well. So on to vaccines. I'm going to be spending time talking. I was going to talk about 3, but given I only have 10 minutes, I'm going to limit that to 2. different vaccine candidates. But the three that are furthest advanced in clinical trials and approaching phase 3 efficacy trials are shown here diagrammatically. First on the top, you see the dengue genome. There are about 10 gene products. The important gene products here in terms of antibody responses are the external proteins, the PRM and envelope proteins. And then important targets for cellular immune responses are the non-structural genes to the 3' end of those. So what various folks have done is taken the genome and essentially constructed live attenuated dengue virus vaccines. So these are all live attenuated viruses, and so don't require adjuvants, but just simply infect and generate a good immune response. Sanofi Pasteur uses technology that was developed at a campus where they took a yellow fever virus and transposed then the membrane and envelope proteins from that yellow fever virus from each dengue virus into the yellow fever virus to create four different chimeras. So these chimeras will generate antibody responses and hopefully neutralizing antibody responses to each of the 4 viruses, but their cellular immunity to the non-structural genes will be limited to yellow fever. The vaccine that Invirigen worked on and now Takeda is developing was actually invented at the CDC. We licensed it from them, the CDC in the US, where they took an attenuated and well-characterized dengue 2 strain, which then has the non-structural genes of dengue 2, of course. It has mutations, several attenuating mutations outside the structural genes, outside PRM and E. So that allowed them to then transpose the structural genes for dengue 1, 3, and 4 into this dengue 2 backbone. Another vaccine that's approaching phase 3 clinical trial was developed at NIH, where they took the dengue virus and made deletions on the 3' end, which attenuated the viruses for 1, 3, and 4, and then they had to make a chimera for dengue 2, transposing again the PRM and E streams from dengue 2 into this dengue 4 backbone. So the differences here are in terms of the cellular immunity. All potentially can develop neutralizing antibodies against the surface proteins. Our vaccine will develop cellular immune responses to the dengue 2 backbone, and of course the non-structural genes are very conserved between the dengue viruses, so we're relying on those conserved non-structural epitopes then to generate cellular responses against all 4, and we have seen that in monkey and now human samples. NIH then has 3 different backbones, dengue 1, 3, and 4, all capable of generating the whole plethora of immune responses. So where are we in terms of clinical testing of these live attenuated vaccine candidates? The furthest along is the Sanofi vaccine, and what was a highlight then of last year was when they published 2 phase 3 efficacy studies of their vaccine candidate. Prior to that, there was also a phase 2b efficacy study in which there were 4,000 children in Thailand which were vaccinated. And then the phase 3 first published in Southeast Asia was about 10,000 children aged 2 to 14. And then in Latin America, they immunized 20,000 children ages 9 to 16, and that was the most recently published. In their vaccine requires 3 doses to generate immune responses to all 4 viruses, given at 0, 6, and 12 months. And then they followed up for a year then to see which kids got dengue fever and which did not after the 3rd immunization. So the primary endpoint was protection against dengue fever caused by any virus. And that primary endpoint was met in their 2 phase 3 clinical trials. So this is a hallelujah for the field. I mean, here we actually have our first dengue vaccine that shows clinical efficacy. The efficacy was reasonable. It was 56% in the Southeast Asian trial, about 61% then in the LATAM study. The other important highlights, though, of the publications here was that not only did they prevent protection from dengue fever of any cause, so what they're doing is taking fever in any child and then diagnosing dengue by PCR, okay? So it's fever of any severity. But they also actually protected against severe dengue, hemorrhagic fever, shock syndrome, or other more severe consequences. The numbers are small, but they're still significant, in 80 and 96%. And they also showed an effect on hospitalization due to dengue. Again, efficacy of 67 and 60% in the 2 trials. So based on the strength of these data, it's likely that this is going to be licensed this year, which is very exciting. That said, there are some unusual aspects of the protection engendered by this vaccine, which I'll review here. Here are the data then on the 3 different trials, the phase 2b and then the 2 phase 3 trials, showing the efficacy endpoint the point estimate, and then the 95% confidence intervals. Their phase 2B that was done in Thailand actually failed to show significant efficacy. The overall efficacy was about 30%, but the 95% confidence interval overlapped 0. And I'll tell you why in a minute. Their Asian and Latin American trials, however, again, 56 and the 60% efficacy with nice narrow 95% confidence intervals, so clearly significant protection. So one of the reasons then, though, that this Thai phase 2B failed was because they saw very different degrees of efficacy for the 4 different viruses as shown here. Again, the point estimates and the 95% confidence intervals. This is now dengue fever caused by dengue virus 1. And what they found, about 50% efficacy in all 3 trials. Again, better confidence intervals, of course, in the larger trials. So that's modest efficacy, but not terrific. Dengue 2, however, was a big problem. Here they saw efficacy of only 35% and 40%. The 2 large phase 3 clinical trials is only 9% in the Asian study in the phase 2B done in Thailand. And the other problem of the phase 2B done in Thailand was that 50% of the cases were dengue 2. So you can see here that that one virus, the lower efficacy with that one virus could then bias their results to the lower levels of efficacy seen here. Efficacy against dengue 3 and 4 are in the 70s, so 70 to 80% efficacy for both dengue 3 and dengue 4 in all 3 trials, and again, much better numbers with the larger scale clinical trials done in multiple centers in Asia and then in Latin America. Another interesting wrinkle in terms of the efficacy is if you look at those individuals who, upon enrollment in the trial, had either been pre-exposed to dengue, or dengue seropositive, or were naive to dengue. So these are the younger children who never seen dengue before, had not been bitten by a dengue-infected mosquito. And those are, whoops, sorry about that. Those are seronegative here. And in the Asian trial, you can see very good efficacy, 75 to 80% in the seropositives. But the efficacy then in terms of seronegative individuals, only 35% and 41%. And again, the error bars are large here because one of the other weaknesses of the Sanofi trial was that they did an immunogenicity subset. They didn't look at immune responses in every participant because you're trying to enroll lots of children into these trials and you don't want to be taking a lot of blood samples. So the immunogenicity subsets were restricted to only 2,000 subjects each. And of course, identifying these seropositives and seronegatives requires those subset analyses. Then analogously, if you look at efficacy by age, again, the younger kids who are mostly seronegative also show lower efficacy than the older children. So this presents sort of a challenge in terms of how one might then actually use this vaccine in a public health perspective that we'll get to at the end. So on to where Takeda is with the Envirigen vaccine based on dengue 2 I was telling you about a few minutes ago. We've completed 2 phase 1 clinical studies and a phase 2 clinical study. The phase 1s are published, and that was done by Sarah George as the principal investigator in St. Louis in the USA, where dengue is not endemic. But we also did a study in Colombia, actually at high altitude. Colombia is a dengue endemic country, but at high altitude, about 7,000 feet above Medellin, in Rio Negro, actually, which is close to where the airport is, and for those flying into Medellin, there's no dengue because the Aedes aegypti can't survive the cold temperatures at night. And so we did a study up there with Yvonne Velez as our collaborator from PSET. And both were randomized placebo-controlled study. They were similarly designed. Both had 2 different dose formulations of the tetravalent dengue vaccine, TDV, and both looked at both intradermal versus subcutaneous immunization. And again, healthy, and they were screened to be flavivirus negative before enrolling into the study. The completed phase 2 that we've done is multi-center, was done in Singapore, in Thailand, and Puerto Rico, and Colombia, and looking at both adults and children now in dengue endemic settings. The data from the phase 1 clinical study were quite encouraging. It was quite safe. We saw no serious adverse events related to vaccine. Interestingly, no other hallmarks of dengue, such as rash or reduction in platelet counts. No vaccine-related adverse events. Most of the systemic adverse events, most common were headache, colds, nausea, and myalgia, but equally common in the placebo groups, and no constellation of symptoms that were like dengue, as I mentioned. What we did see was local site reactions, which would be what one might see with an active live viral infection. So erythema and some edema at the injection site. We saw more erythema and edema with intradermal than with subcutaneous injection, and those were short duration. We didn't see any changes in hematology or blood chemistry, and no discontinuations, no death. How about immunogenicity? And here, the pattern we see with our vaccine is very different than Sanofi saw with theirs. They optimized theirs to get even responses to all 4 viruses in terms of neutralizing antibodies. What we see is we see higher levels of neutralizing antibodies to dengue 2, followed by 1 and 3, and then the lowest levels to 4. So we actually hope that these higher levels of neutralizing antibodies may help us in terms of protecting against the more pernicious dengue 2 virus in the future. But what about generating responses against all 4 patients, all 4 viruses? I mentioned that the Sanofi vaccine required 3 doses. We see in 1 or 2 doses very significant tetravalent immune responses. So this shows seroconversion to either 4 viruses on the left here in each of the graphs, or 3 or more viruses, 2 or more, 1 or more. And you can see that even 1 dose generates significant levels of tetravalent immune responses. That's increased slightly by the 2nd dose. And you see large percentages of trivalent or more immune responses. That's important because natural infection with 2 viruses can lead to protection against the 3rd or 4th as well. So we're very encouraged by these immune responses. And moving forward then, we had another phase 1 that's been completed now, looking at different schedules and some more dose finding. And in addition, then we have 2 important studies expanding the database for the vaccine in order to prepare for phase 3 that are ongoing right now, looking at 1,000 dengue naive subjects and our final formulations that are going to go into the clinic, and then also looking at safety and immunogenicity, again, in dengue endemic countries and kids, 1,800 subjects in this case, so significantly expanding our safety database before we launch phase 3 clinical trials this year. So very encouraged. In the phase 2, we saw greater than 80% of the subjects being seropositive to all 4 viruses after 2 doses. So what's going to happen for the rest of the year in terms of dengue vaccines? I think it's going to be a great year between now and the next D3 meeting. First of all, Sanofi. Sanofi is going to get approval for their vaccine. Their data are robust. They've submitted now to regulatory agencies, and they're just waiting to hear. But that's still not the end of the story for them. First of all, with these dengue vaccines, it's very important to do long-term safety follow-up, and that's ongoing for the phase 2B and the 2 phase 3 trials that Sanofi has described. WHO guidelines require at least 3 years of long-term follow-up because you worry if the immune responses wane, are you then subject to more severe disease? So those are very data that will be forthcoming. In addition, of course, how will it be implemented? If right now Sanofi, because of that increased efficacy in seropositives, is targeting 10 to 11-year-olds as potential first children to be vaccinated. With very little efficacy in the youngest children, that means at all times there'll be a population that'll still be subject to dengue. So that may be a significant problem going forward. Important for us developers of vaccines after Sanofi is, will there be a correlative immunity identified? And there, the limited immunogenicity subset may be a problem. And then importantly, if the Sanofi vaccine is implemented by public health authorities, how is that going to affect future efficacy studies? If it's standard of care, it has to be the placebo. So that may then inhibit the efficacy studies by others in the future. But as I mentioned, Takeda is preparing for a phase 3. The NIH vaccine, which I did also want to talk about because it's a very good vaccine candidate, is moving forward as well. They've done 2 phase 1s with admixtures, and they have a phase 2 underway in Thailand on phase 1 and 2 studies. But one of their licensees, Butantan, have also now started with a tetravalent formulation, and Butantan is intending to do a phase 3 in Brazil also fairly quickly. So it's going to be a great year in terms of Sanofi rolling out their vaccine, and then 2 other vaccine candidates with phase 3 clinical trials ongoing. Last but not least, another important aspect of dengue vaccine development is that now people are developing human challenge models to more carefully look at the correlates of protection in more controlled situations, and those will be really valuable data on in the future. And beyond dengue, of course, there's still chikungunya. So I'll leave that for the panel discussion. Thank you.
Thank you very much, Dan, for that.